because the culture is different and it's always rainy and foggy and it looks more like England. So if you come here, because some people, they apply for postdoc and they say, yeah, I want to go to Spain, and then they come here and they see the rain and they get disappointed. So you are forewarned. Here is more like Ireland than in Mediterranean, okay? And Achucarro is a very young institute. It's like 7 years old. We have like 18 teams, but most of them are very small. We are a public research center. We have, in particular, good imaging facilities. In the rest, we are okay, but the best thing that we have is probably the microscopes. And we are a center oriented to the study of glial cells, which I will explain later because I know that some people are not biologists, so I will have to introduce the glial cells. So typically, I don't know if you see my cursor here. I mean, normally we have the neurons. I mean, the people are most used to, when they study the brain, to think about the neurons. But here in Achucarro, and most of the work I will present today at the end of the talk, when I present our results, is about the glial cells, which are basically, there are more, but the basic ones are astrocytes, oligodendrocytes, and microglia. So today's lecture, we have a plan. Basically, I will start doing an introduction to fluorescent microscopy. Then I will have to explain the difference between wide field and confocal, even if the talk is about 2 photons, is of paramount importance that you understand this difference, because otherwise you won't understand why we need to use 2 photons. Then we will explain what is 2 photon microscopy, and this is the technical part, more about physics. Then the pros and cons of 2 photon microscopy, the applications, for example, like calcium imaging. Then we have some unpublished data about the microglia motility. And then the key points to elaborate the protocol that we have to do after. So fluorescent microscopy, I mean, we use fluorescent microscopy because the brain, normally we want to see at the brain in a live tissue. So when the tissue is alive, so we can see the cells moving and the cells reacting to things. In this case, you can see the microglia, which are reacting to a laser lesion. This is an iconic image from the lab. I always put this image in the beginning of the talk because it represents many things. Not only that the cells move, but that the cells can react, and that you can, with the fluorescent microscopy, you can isolate them, because actually this is a part of brain tissue that is full of cells, is full of extracellular matrix, for example, but you can only, here you're only seeing the microglia. So why we need fluorescent microscopy? We need fluorescent microscopy because it will provide us with these 3 concepts: scale, location, and non-destructive analysis. And I will explain this a little bit more. The scales is because important biological events occur at micro and nanometric scales. So we need to see things that are very tiny, and fluorescent microscopy enables us to see this. Also, we can obtain information about how things happen, but also where, because we can understand how things happen if we do a Western blot or we do a PCR, but to see where it happens, we need the microscopy. We need some microscopy to the analysis. And also the biological organisms are adapted to light. So we can use light to analyze them without destroying them. So this is when I was talking about non-destructive analysis. So with fluorescent microscopy, we can image live samples. And this is more or less explained in this next slide. We have basically 2 types of microscopy. One is electron microscopy, which gives us a lot of resolution. We have two types of electron microscopy. We have transmission electron microscopy, where we pass electrons through the sample, a contrasted sample, and we can see at nanometer scale, for example, the vesicles in the synapses. And then we have the scanning electron microscopy, where we have some kind of like the reflection of the electrons, so we can see the shape of things. Okay. And then we have light microscopy, which doesn't use electrons but use photons to illuminate the tissue. And we have the typical optical microscopy, What we can see here is the hematoxylineosine staining. And we have fluorescent microscopy, which is the one that we are focusing today. Why? Because the other three methods, both electron microscopy and the one from optical, normally they use, with electron microscopy for sure, but with optical microscopy, normally the tissue is fixed. So the cells are dead, so you cannot see dynamics. If you want to analyze dynamics in real time, you need to use or bright field or fluorescent microscopy. A brief history. I'm not going to bother you with all the history of microscopy, but basically the first microscope was invented in the 1600s, and the first microscope they used to look like this. Then we had the microscope that, for example, I used to play with when I was a child. And now a confocal microscope from the 90s or the beginning of the century, it looks like this. And usually the microscopes now, they are much more complex. Normally they are custom made. They have very customized parts to achieve maximal resolution or maximal intensity or whatever. And normally they occupy a full room with several monitors and screens, and normally they look like this. Okay, in this case, is what we call a setup. It's not a microscope anymore. It's like a setup because it has different devices attached to it. Not only the microscope, but also electrophysiology or optogenetics or different kind of possibilities. The two-photon microscope was invented in the 90s. Actually, there is further down the talk, we will see the history of two-photon microscopy, and we will see that it started in the 30s actually. The first two-photon microscope was invented in 1990. And why, with fluorescent microscopy, I said that it enabled us to see living things. So you can see with electron microscopy, we can reach further resolution to see small molecules or lipids. We cannot do this with light microscopy because we have the diffraction barrier. We will talk about this later. But now with some methods of super resolution, we can see even further in the nanometer scale, and we can even see viruses. We're not focusing on super resolution on this talk. Actually, there is another talk about this that we used to do at the master. But just to mention that the super resolution methods, they received the Nobel Prize in 2018. Because as I said, they enhance the resolution that we can observe live with, and also in living samples, right? Because this is the difference, the main difference with electron microscopy. So a few key concepts of light that will be useful for later. Light suffers refraction, and this is important because light can be bent when it goes through different materials, like for example, the water from your sample or the glass from your cover slip or the mounting media. Also, the light can be reflected, and this is something that is very useful, not for two-photon, but for some types of confocal microscopy. So when you want to see, for example, myelin or organized structure, you can use the reflection. Also the dispersion, and this is important because as you can see, when we have white light, it goes through a prism, through some organized pattern. The light of different wavelengths will bend differently, and this means that the resolution we will see later is also dependent on the wavelength. And also the polarization, which is not really important for this talk. Actually, we use it in the two-photon microscope to regulate or modulate the intensity of light. But this is a very technical part of the microscope that we won't be covering today. Also, another note about the fluorophores. This part, I'm sorry for the people that are already working in biology, but I had to make these basic statements because some people they are not used to work with this with fluorescence. So in fluorophores, we basically have two categories, let's say. We have small fluorophores, which are very small molecules that, for example, like Alexa fluor is one of the most used green fluorophores. Tetramethylrhodamine, which is one of the very famous red fluorophore. And this is a fluorophore. It's a green fluorophore but it enhances its fluorescence when it's bound to calcium. So we use it as a calcium indicator. A particular thing about all these molecules, and this will be useful for the next slide, is that if you see these are organic structures, they have double bonds here, and these double bonds are paired with single bonds. So all this structure makes like a flat surface full of electrons above and full of electrons below. So this makes a good like resonance system, and this is how the fluorophores work. So the photons will excite the electrons here that are at the lower energy level because they are very comfortable in these resonance systems, and they will excite them, and then when they relax, they will emit the fluorescence. We will explain this with the diagram down below. Another type of fluorophores that we have are the fluorescent proteins, which are usually worse, and they have less yield. They give less light than the small fluorophores, but they have the great advantage that we can codify them genetically. So, for example, we can put them in a virus, and we can inject them whenever we want in the brain, or we can have a transgenic animal that will express, for example, green microglia or green astrocytes or red neurons or whatever. There are many fluorescent proteins, but the ones that we use normally they come from the GFP, which is from the algae, the green fluorescent protein, and it has many derivatives, like for example the calcium indicator GCAM, we'll see later, comes from the GFP, and also the DS red and all the derivatives like M cherry, M tomato, M banana, like a lot of fluorescent proteins that they have different properties, but they are all modifications of the DS red. And to finish this introductory part about fluorescence, and normally when we want to do a diagram of the energy of the fluorescent event, we use this diagram that are called the Jablonski diagram, where we excite an electron that is in a comfortable state, like I said, in a resonance system. We will excite the photon will excite the electron. The electron will then have a thermal decay, so we release some of energy in a form of heat, and then it will release the rest of the energy in form of fluorescence while it goes back to what we call the dark state. This is the dark state, and this is the fluorescent state. So this means that normally we excite with the wavelength and the photon that it emits, since it lost some of the energy as heat, it will emit a new photon, but with a wavelength that is shifted to the red, we say, because this means that the light will be far on the red in the spectrum. So here, for example, we have the excitation, for example, at 500 nanometers, and the emission will be shifted. This is what normally happens. We will see that in the two-photon phenomena, this is different. And the concept of the diffraction barrier, that I was telling you before about the super resolution. The problem with the diffraction of light is that the light, when it passes through small gaps, like the ones between the molecules, it is bent. So since light is bent by these small molecules, and the gap between the molecules is important, resolution of light microscopy is limited by the Abbe's equation. Normally, this limit is about 200 nanometers for green fluorophores. The equation is summarized here. It depends on the wavelength directly and indirectly from the numerical aperture or the resolution of the objective, let's say. Okay, I'm missing a C here, but this depends on the wavelength and the numerical aperture. And this is why normally when we want to see things with high resolution, we need a 1.4 numerical aperture fluorophore. Give me a minute. Okay. So then the diffraction of barrier means that when we have a real object, normally the light will be distorted in a way that when we observe the object, the object will be blurry. And this is what happens in the XY plane, and the Z plane it will be even worse. And this deformation, let's say, of the light from the object is called it follows a mathematical function that we call the point spread function. And this will be important for the resolution that we will have later in two-photon. Because in two-photon microscopy, let me go back a little bit to the equation. This wavelength is very high. It's like 900 nanometers, not 400 or 500 like we normally use in the lab. In the two-photon, this is very high, so this means that it will lower the resolution that we can use. So this PSF can be overcome with two methods, basically. One is with super resolution. The super resolution will enhance the PSF, or with deconvolution. The deconvolution will calculate mathematically a better PSF from like a theoretical PSF from the real PSF. So this is just to tell you that fluorescence is not perfect. Fluorescence has problems of resolution that we can overcome with different modalities. Okay. So before going with the confocal and the two-photon, so fluorescence allows us to image living brain tissue with fluorescence. And for example, in this is an organotypic culture where we have this is the NCAN GFP, and NORCAN GFP is a living a label of the extracellular matrix. And the Alexa 594, which here is in magenta, is tagging microglia with isolectin. So here we can follow the cells. You can see with fluorescence, we can see things that move like the microglia and things that don't move like the matrix. But we have a real-time map of both. So normally you have to do an analysis. Here we are separating the channels. We have the channel of the matrix. We have the channel of microglia. We can segment this microglia, and this is an important part of the analysis that we will see at the end of the protocol. And we can track them, so that we can have a map of microglia's movement in the context of the matrix, for example. So how can we get sharper images? The typical answer normally is that we go to a confocal microscope. And in a confocal microscope, we are able to see, we can convert a blurry image to a sharp image in the confocal. So this is the part where I will explain more or less the differences between wide field and confocal, because the two-photon is very similar to the confocal. So you have to understand this difference first. So in wide field, it's like when you take a picture with a bokeh or your portrait mode in your iPhone or your Android. No, you have focus and out of focus information. And in fluorescent microscopy, sometimes this is too much because we just want information from the focal plane. So in the confocal, normally you filter most of the light. So you eliminate out of focus light by using what we call the pinhole, and you get only the information from the focal plane. Two-photon does the same, and we will see later the difference between confocal and two-photon and why sometimes we choose two-photon. Okay, so when we build a microscope, the difference in wide field and in confocal, basically the excitation source are different. We will see a table later when we compare both. But the main difference is that in the confocal you have the pinhole, and this pinhole will filter most of the light, all of the out of focus light. So you will get only the light from a focal plane. So you will do what we call the optical sectioning. So it's like you cut only a thin slice of tissue. You can image only this part, and this is how you get the sharpness. So there are big differences between wide field and confocal. For example, the light source in wide field, you can use LEDs. We used to use mercury lamps and xenon lamps, but now we don't use them anymore. We use mostly LEDs. But in the confocal, you need a laser. And why you need a laser? Because you're filtering most of the light. And this is important because if you understand this, then you will understand why we need to use two-photon for some application. So you use a laser because the light that you collect is very few because it's filtered through the pinhole. So it means that you have to provide to the system much more energy, much and this is why you use the laser, so you can collect at least something through the pinhole, right? So this is according to the light source. Then the separation of the emitted light. Normally in wide field you use normal dichroic mirrors and filters. We will see them later. In confocal, this can get more complicated sometimes because you can use this very nice acoustic-optic filters. They give you a very narrow band of light, so you can clearly separate, for example, I don't know, 500 nanometers from 510, usually. The detection normally is done with cameras, and in confocal is done with photomultipliers because you have to amplify the light. In wide field, you get a lot of light, so you can use a camera. And a camera means that you can be fast. Okay, when you have living things, normally you need to be fast, and you can use camera. You have to use cameras. In confocal, if you use cameras, you won't get enough signal because it's too few light going through the pinhole. So normally you have to amplify it with the photomultipliers or with the hybrid detectors, semiconductors. Different. The way of collecting the light is different. The image acquisition is also different because in wide field you take an image to the whole sample at the same time, and in confocal you have to scan the field. So in confocal it will go through all the field point by point. This is why it's called point scanning, and this takes time. This is why it's slow. In wide field you have no pinhole. In wide field you can't do 3D reconstruction unless you do the convolution first. Normally in wide field you get a lot of light, and in confocal you get not much light, but you have much sharper images than in wide field. And this is critical because acquisition speed in wide field is really fast, and in confocal is usually slow, unless you have a spinning disk microscope, which is a different type of confocal that I won't be covering today. So the applications, and this is the important part. In wide field, you use it for live imaging, for fixed cells, but in vitro because you don't have sharpness. And for structural illumination, this is something for super resolution that is not useful now for us. Confocal normally is used for fixed tissue. You can use it for live imaging, but you will photo bleach your sample probably. And for colocalization microscopy and for other super resolution methods. The problem is that I told you in the beginning of the talk that we need to do live imaging because we want to see dynamics. So this means that we need we need something. We need the best from both modalities. We need the sharpness of the confocal and the speed or the way that wide field takes images from live samples. So I think in the next I have, yes. So this is a comparison between wide field and confocal. In wide field we illuminate a lot of the sample and we get a huge PSF. So we get focus and out of focus light. In confocal we get light only from the focal point. And the colors here, they matter because normally you illuminate with green, you will get red light. And if you illuminate with green, you will get red light too. In two photon, it's the inverse. Normally we will illuminate with infrared light. And with infrared light, we will get green light. And this is one of the keys of the technique because illuminated with red light, and especially with infrared light, will give us depth. I mean, we will be able to go microns below the surface of the brain, microns below the surface of the acute slice, as we will do in the protocol at the end. Basically, we also only illuminate the focal point, and this is another critical point. And in this figure, there's everything. I will explain it more detail later, but here you can see that first the shift is from red to green, so from low energy to high energy. And also the excitation is only at the focal point. So here is an example of one photon excitation. Normally you illuminate the whole cone of light. So you illuminate the whole sample, but then you will collect the light that comes from this point in the middle. That is the focal point. In two photon, you only excite this part. And this is the trick. So you are giving to the system much less energy. So how two photon excitation works? So this is the Jablonski diagram, a little bit more fancy, but it's the same as before. You excite, for example, with blue light and you receive green light. in the 2-photon, the electron or the fluorophore, it will absorb 2 photons of lower energy, in this case 976 nanometers, or 920 or 820. I mean, we use different wavelengths for different fluorophores. But basically, you will excite 2 with 2 photons at the same time. This is why it's called 2-photon. So they will sum their energy, and it will be as if I were exciting with only 1 photon blue, only 1 blue photon. Here I'm using 2 red photons, okay? But I will get the same result, green light of 510 nanometers. But the energy I'm giving to the system is half. So this is one of the important points. The problem is that for the 2-photon, I will need a very special laser, and as I will explain later. The configuration of the microscope is the 2-photon is similar to the Y-field, so I have no pinhole. The difference is that the light, the emitted light will only come from the focal point. So this will be the configuration, no pinhole, so the system is simpler. So 2-photon excitation generates fluorescence only on the focal plane, as I told you. But the important thing here is the laser, and this is what makes the microscope expensive, and this makes it different to other typical microscope, because the lasers are expensive and are hard to maintain. I mean, you have to cool them with water. I mean, it's a complicated laser. So basically, the laser from the 2-photon microscope, we will see that now we have more types of lasers. But it's a laser made with a crystal of titanium sapphire. And it's called the femtosecond laser because it will emit a pulse of, I mean, like a pulse of millions of photons, but at pulses that have the duration of 1 elevated to 10 and minus 14 seconds. So very short pulses. So these pulses are so short and are so sequential in time that you are sending like a huge amount of photons to the same place, and this is what happens here at the focal plane. So I'm sending a lot of photons to this tiny spot in the middle. So I'm sending so many photons that that the chances of 2 photons of colliding and exciting the same fluorophore, the 2 at the same times, is much higher than in the rest of the tissue. I mean, there is light in the rest of the plane, in the rest of the tissue, sorry, but there are not enough photons in the rest of the tissue. The concentration of photons is higher at the focal point, and it's only at the focal point when I will have the 2-photon phenomena happen. And this is the key, because everything will happen here because I'm using these super fast, super short pulses of the femtosecond laser. So this is the key, that the 2-photon microscope has the pulses that last the femtosecond, that we can tune them. Normally, these are the titanium sapphire are tunable lasers, so I can set them to different wavelengths to excite different fluorophores. And the photomultiplier tubes are similar to the ones in confocal, made of gallium arsenide and phosphide, the GASP we call them. And we can align them serially using different filters. As you see here, normally here you have the femtosecond laser is red, the light is red, we excite, we collect, for example, yellow, green, and blue light. And some light is going to one photomultiplicator and some light is going to another photomultiplicator, for example. We can do this normally, and we will see this at the end when we present the protocol. Because the configuration that we have in both our microscope here at the Chúcaro, they have this serial photomultiplicator. So the key thing here is the laser, that is pulse laser. So this is what I was talking in the beginning with Jorge about the new type of laser that we have now. This is the laser that has been used for, I don't know, 30 years in 2-photon, is the titanium sapphire laser, which, as I said, is tunable, is large. It needs water cooling, it's very expensive, and it has very short pulses, and the power is very high. Now they have this, but normally for biology, you don't need that much power, okay? So for biology, you can use these new ones, which are called the fiber-based lasers. So the lights go through a fiber, and this fiber is doped. So since it's doped, it gets interference from the dope. The dope is like a contamination in the fiber. So this interference will create the pulses. In this case, the interference will be created by all the rebounds of the light between the multiple mirrors. Sometimes the light will collide with itself and will create the pulses because of the optical path. It's very complicated, but the key thing here is that these lasers are much cheaper, but really much cheaper. I mean, the price of a laser like this one is about 200,000 euros, and this one is 40,000 euros or 30, much cheaper. The pulse duration is acceptable. The power is really enough for biology. The only problem is that they are non-tunable. They are at a fixed wavelength. So if you want to do calcium imaging, you have to buy one. But if you also want to do, I don't know, red neurons, you have to buy another laser. But with two lasers of 40,000 euros, you can have a perfect two-photon microscope. Okay. So this is why I was saying that it's getting cheaper and it's getting close to the price of the confocal. And the last concept is to understand the applications of two-photon is that in two-photon the excitation is pulsed because it's obligatory. We need to get pulses so we can get all those photons in this is a more small space and time. It means that even if the power of each pulse is very high, the average power, I think I have here, yes, the average power is much lower. While the continuous wave, which is the laser that we normally use for confocal, the average power is much higher. So you are giving much more energy to the system. This is important for living cells because they get burned, literally. So why two-photon microscopy? So with all I explained you until now, two-photon microscopy will give us lower average power. So this means reduced phototoxicity, and this is key. For example, I study microglia, and if you give microglia a lot of light, it will become reactive. So it's not a question of using, you can use confocal, but you will have problems. And if you use calcium, for calcium imaging, you will probably bleach your fluorophore, so you are forced to use two-photon microscopy. Another advantage is that you have increased imaging depth, so because of the near infrared excitation, and you have less scattering on this wavelength, so you can go deeper. Normally with the photon, I can say, people say that they can go to 350, 400 microns, but I've never seen this. 200 microns at the most. You can go deeper, but you have to increase the power so much that you will hit the brain, okay? And you will cause swelling. You will cause swelling, so it's not possible. And this is very important too. The excitation is spatially restricted, so you will excite only 1 femtoliter in the focal point, and this is very useful for biology, as we will see later in the different applications of two-photon. The milestones in two-photon microscopy. I just wanted to point two dates here. First, that the Nobel Prize winner Maria Gopper-Meyer, she described theoretically the concept of two-photon in 1931, and it was not until 60 years later when they developed the first two-photon laser scanning microscope at the University of Cornell University. And the first calcium imaging was done in 2003, and the first microglia motility, very famous paper by Alex Nimerjan, was done in 2005. So from the development to the usage of the two-photon for the first time in the brain, it was more than a decade. So applications, and I will explain these 5. We will leave ex vivo imaging for the last because it is the one that we will use in the protocol, the one I do almost every day. So we will explain the other 4 first. So in vivo imaging is probably the main application of two-photon, and most people, they use it for this. We use it for this too, but it's not our everyday usage. Okay. So in vivo is pretty complicated, but not the microscopy itself, but the surgery. You have to do a very special surgery. Normally we do this on mice. I've seen it in rats, but in mice it's much easier because they are small. Normally you have to open the skull of the mouse. We call this the cranial window. So normally you have to put some, you have to glue to the skull some kind of reservoir so you can hold some artificial cerebrospinal fluid. This ACSF is a liquid that resembles the cerebrospinal fluid, the real cerebrospinal fluid of the brain. Okay, and normally you have to use some agar, or you have to put a cover slip so the brain doesn't get infected, and then you have to place the objective just on top. And the problem here, the mouse will move, even if it's, you can do it with anesthetized mice, or you can use it with awake mice. But the mouse has to be fixed. So normally you have to use like a restraining device, or normally we put what we call the head plate, which is this tiny, in this case they use a ring, we use a different device, but it's similar, it's basically same, serves for the same purpose, that it will fix the head of the mouse to the objective, and you will have a still image of, for example, a neuron, and you can image very deep here. This is a transgenic mouse, it's not a virus or GCAM or whatever, but you can go very deep, even up to layer 4 or 5, okay? This is an experiment that we did in Achucarro in 2022, I think we published it later, but this was calcium imaging in vivo. So here what you're seeing, this is astrocytes, okay? This is GCAM6, which is a calcium indicator, fluorescent calcium indicator in GFAP Cree mouse. So the GCAM is only expressed in the astrocytes. So here, Juliana from Toledo, Juliana Rosa from the Hospital Nacional de Parapléjicos in Toledo, she came to Achucarro to do this experiment. She put the electrodes on the paw of the mouse. So she was stimulating a specific region. So she wanted to see how the signal arrives to this point when you see the laser here, and then the wave of calcium spreads to the rest of the astrocytic network. So she was trying to prove how astrocytes process information from in the sensory motor cortex, right? So this is the key application, calcium imaging. This is in astrocytes because it's the one we do, but you can also see the neurons. You can also, now people are using it also for voltage. So they're doing like electrophysiology with light, okay? But the main application of in vivo to photon is for calcium imaging. This is some of the quantifications, how you see the astrocyte and the different traces of calcium. And this is normally for neurons, they are different because these are wide because there are astrocytes. For neurons, they are much shorter. But basically what you can do is you can see the response of the brain of a systemic stimulation, in this case electricity in the paw. Also is being used, this is not from our lab, this is from a lab in Paris, that they use this probe. It's called the laconic. It's a FRET probe. FRET is fluorescent resonant energy transfer. So it's a probe that when it is bound to lactate, it will change its fluorescent pattern. Let's say, typically this, you will excite this part of the fluorophore, which is the TFP, and it will transfer the energy to this yellow part. So you excite this one, but then you collect the light from this yellow part. So I don't remember how it works exactly, but I think when you bind with lactate binds to this, it will enable the FRET, and when you have no lactate, there is no FRET. So you can quantify the ratio between both, which is this ratio that you normalized MTSP Venus, and you can see these small changes in astrocyte and in lactate uptake, and to see in real time the metabolism of different cells in the brain. Just to show you something that is not calcium, okay? And also you can see structures. In this case, there's no calcium, there's no lactate. Here is dual color to photon microscopy. In green, this is from the lab of Martin Furman in Bonn. Here he has in green, he has the microglia, and in magenta he has the spines. So here he can image in vivo in the hippocampus, and this is very difficult to do because he has to basically implant the objective in the hippocampus. he can analyze the dynamics of microglia in relation to the dendrites. So I think I don't have the quantification. I think I have it in another presentation. But here what he saw is that microglia touches the spines all the time. And if it touches a lot of times, the spine will disappear. And if it touches less times, the spines will be kept alive, let's say. So the growth and retraction of a spine depends on the number of contacts by microglia. And this is only possible with dual color to photomicroscopy. So another application, and now we will use what I told you about the spatial restriction, that we can excite a tiny part of the tissue, is for example to do glutamate uncaging. Glutamate uncaging means that we will have glutamate, which is a neurotransmitter that is trapped by another molecule. And we can break this link using light. So when we apply the light, we will break this bond between this modifier and the glutamate will be free and will excite the spines. So what happens if you want to excite this spine, but you don't want to excite the rest of the dendrite, and you want to excite the spine that is next to it? So you need a spatial precision. And this spatial precision is only possible with the 2-photon, because if you do it with the confocal, you will excite like a lot of spines, the ones that are above your sample and the ones that are below your sample. So 2-photon is it's been used for years for this uncaging experiment. Here they are exciting this spine, and they can see how it changes the plasticity with at different time points. I mean, the important here is, I mean, you can do it with in this case with the spine, but people, they use it also at the synapse and the pre-synapse and the post-synapse. I mean, you can do different experiments with this because the 2-photon will give you the spatial precision necessary to this type of experiments. Another important application is the in situ ablation. So you can do localized damage, which is the video that I showed you in the beginning with the microglia. Okay, in this case, I will present you another example. It has nothing to do with microglia. This is in zebrafish, so it's in vivo, but it's what I like to say the easy in vivo, because in zebrafish, I mean, the system is much easier. You don't need the head plate or the cranial window or nothing, because the zebrafish is transparent. So here they have the oligodendrocytes. Some oligodendrocytes, they have them. All oligodendrocytes, they have this killer red, which is a fluorophore that, when excited, it will generate free radicals, and it will kill the cell. So they have 2 oligodendrocytes. This one, which all the myelin sheets here, and they have the number 1 and number 2, and they only kill this oligodendrocyte. They leave this one alive, and they can see how this oligodendrocyte take the task from this one and will myelinate the axons that were left unmyelinated by this one. So they can see in real time how one oligodendrocyte take the task of another one. This paper was interesting because it happens really fast. Normally in the mouse brain you have to wait days to happen this, but in zebrafish they could already see that some myelin was forming really fast. So another application that I didn't used to cover this, but I included here because it's interesting for the people that work in the matrix like me, is that with 2-photon and not with confocal, you can use a strange phenomenon, or strange for me because I don't understand completely, called second harmonic generation, which is a different phenomenon from 2-photon absorption. So here is the explanation. In 1 photon, you have typically you have the light will excite and then will relax and then will emit the fluorescence. In 2-photon, you have the same. 2 photons will be absorbed and they will release fluorescence of higher energy. In second harmonic generation, there is no absorption. So the light will collide with the tissue, but it won't get absorbed. But and this only happens, I think I put it here, it works only in highly organized structures like myelin, or like, actually myelin is not proved yet. It's something that we're trying to do, but in very organized structure like the collagen matrix, which this is not in brain tissue. I think this is skin, but in very organized tissue structures like collagen, you can have the two photons will rebound against the tissue and will create a harmonic, like in the guitar, for example. When you're tuning a guitar and you create the harmonics, is something that comes from the waves. I'm not a physicist, so it's hard for me to explain it, but when they explained it to me, you have to think, they told me, you have to think, Fede, as in the guitar. You touch, like you create one note, but this will reverberate and create a higher one. So this is similar. Okay, so here there's no absorption, so you are not giving light to this, you are not giving energy to the system, but because you will recover all the energy. And the energy will be exactly the double of the photons of the incident photon. Here is not the same. You cannot sum this and then, I mean, this number is very variable. It depends on the fluorophore because it depends how much it will decay, and this changes for every fluorophore. So the two photon absorption is completely different to this one, and this one only happens, as I told you, is in very organized structure that will generate this reverberation of the wave. Okay, but it's very useful because you can have what we call free labeling. I mean, you can see a structure without labeling it, without a fluorophore, without a virus, without anything. You can, just because the tissue is organized in a particular way. And the last application, and the one that we'll explain, I don't know if I have time. Yeah, we're okay. We have like 10 minutes. Is the ex vivo imaging that we use it. We use the two photon mostly for the reduced phototoxicity, and this is why we use it for microglia. The first example is with neurons, typical experiment with GCAM, spontaneous neuronal activity, and evoked neuronal activity with the potassium chloride. Potassium chloride will depolarize all the neurons, and they will all release calcium to the cytoplasm at once. So what do you need to perform an experiment like this? I think we have 5 or 6 slides left. So you will need a mouse with fluorescent microglia. In this case, we'll do experiments like the one we do in the lab. It's ex vivo imaging of microglia motility. So you will need a mouse. We do it with older mice, but with P20, P30 is easier because you don't have much myelin yet. So the slices, they survive much better. And you will need fluorescent microglia. Oh, actually, it's fluorescent macrophages, monocytes too. But since we are in the brain, most of the green cells that we will see are microglia. Some of them are perivascular macrophages, but you have to be careful and not image close to the blood vessel, and that's it. Or you can have the FMS-GFP, which are the ones that we're using right now from Amanda Sierra, and you can have the red microglia, which is my dream. But I don't have money to buy this mouse, but it will be incredible to have red microglia, so we can combine it with the matrix data. So you need something fluorescent to image. Then you will need to put the slices in a medium that resembles the cerebrospinal fluid. So we will see the recipe of this later, but this is what we call the ACSF, which is artificial cerebrospinal fluid, and the one with sucrose for the slicing and the regular for recovery and imaging. Then you will need the acute brain slices preparing the vibratome. Normally we use 350 microns. And then you will need the 2-photon microscope. And you will need an imaging chamber. We will see it. We will see it in a photo in the next slides. Oxygenated heating perfusion system, because you need to maintain your slices oxygenated and warm. You will need a good objective because you need to have enough resolution to discern the branches of the microglia. You will need some kind of epifluorescence imaging system so you can see where you are before doing the 2-photon imaging, and the filters. And image analysis software, Fiji or Python, and the scripts. So the overview of the protocol, as I told you, is the mouse. You will extract the brain. You will slice it in the vibratome. You will get the slices. These slices, they have to be maintained. This is empty, but it should be full of ACSF. And we bubble it because you need to maintain them oxygenated. This is critical. Then you put them in the chamber, this is a drawing. I will show you a chamber in a minute. So in the chamber you will have the objective. This is it won't be bright light. It will be infrared, as I told you before. But you will need to measure the temperature. You need to have pre-warm ACSF in and take the ACSF out. And it's not a 3D printed chamber, it's a commercial one, okay? Acquisition parameters. The laser should be at 920, which is the optimal excitation wavelength for GFP. This is normally for the green fluorescent proteins is this, 920. For the red fluorescent proteins, it's about 1000 nanometers. And for the small fluorophores like the Alexa is 800. If you have to do this experiment in real life, there are papers that have very long list for all the wavelengths for each fluorophore, so don't worry. The laser at about 50 milliwatts. This is because if you go over 30, you will burn the sample. The PMT normally at saturation. This is something that we don't do for calcium. We only do it for microglia because we want to saturate the signal. We want to see the tiny little processes they have. For calcium, you go to the opposite. You cannot saturate because if you saturate, you won't see the changes. So we do it with saturation because we want to see a structure, not a change in calcium, for example. And the pixel size also about 300 nanometers, so you can see the processes. Normally we do a time lapse. We do a set stack every 1 minute for a total of 30 minutes. We do images every 2 microns, and the total is 40 microns, so it's about 20 images, right? And then the image analysis is long and it takes a lot of time. Normally you have to concatenate the stacks. This depends on your system. Our system gives us the stacks individually. Some other software, they give you the whole time lapse already done. You have to denoise your images or apply some filters. You have to correct the drift, correct the bleach, do a maximum projection or not, depending on what you are measuring. If you're using, for example, Jorge's software, it won't use the maximum projection. And the ones we do for the more common analysis, we use it. Then you will have to segment and quantify. Segmenting means that you will have to create a binary image when the microglia is the signal, okay? And a few more slides with pictures so you see what I'm talking about, because it's very difficult to grasp this without being in the lab. Acute slices, they look like this. You have the brain, you cut it, you slice it, you stick it to the plate with glue, super glue, and then you slice it in the vibrotom. Then you can maintain the slices normally in a chamber like this. We have exactly this one because they allow for very, very low volume of ACSF, and you can incubate another fluorophore like a calcium fluorescent indicator, and then you wash them in a device like this. The slices are kept in this type of net, and here is full of ACSF and it's bubbling, and the bubbling doesn't touch the slices because if the bubbles touch the slices will get damaged, and this is bad for microglia, right? The imaging chamber, this is a picture I found in the internet. Ours is not like this, but you get the idea. Ours is like this one. I mean, you put the slice here, and you have the water goes above and below the chamber, the slice, so it won't disturb the ACSF right where you're doing the imaging. So this is good to have less drift. And normally we hold the slice with something that we call the harp, because it looks like a harp, and this will maintain the slice in place even with small changes of water, of ACSF. And normally in our microscope, because it's microscope for ex vivo, we have infrared illumination and differential interference construct. This is typical from electrophysiology setups, but we have the same in the 2-photon, because in this case, using this type of illumination before using the 2-photon, you will be able to see the neurons, you will be able to see the tissue, and you will be able to see where you are. For example, the people that work in the hippocampus, they want to find the dentate gyrus or the CA1, etc., and they this type of illumination is very useful. So you can see the health of the slice, you can see where you are, and then you go to 2-photon, because in 2-photon everything is black, right? Except the microglia, but here you see everything, so you can orient yourself in the slice. The filters are very simple. You have excitation light. This is a dichroic mirror, so the mirror will reflect green light, and when the red light comes back, it will go through it. So it will go through the emission filter. So you are separating the excitation from emission. This is a typical concept in fluorescent. They look like this. And normally you have to do this kind of diagram so you can understand which type of filters you need. This is the one I use, for example, because I want to separate the GFP or the GCamp from the TD tomato, which is this one. Okay, so in this filter you get this. This is the band. So center is 520, and it has 60 nanometers wide. So 520 is the center, and you have 30 nanometers to the left and 30 nanometers to the right. This is the notation for the filters. And the dichroic mirror will allow light going only on one of the two. Okay. But you can have another filter. For example, there are some people in Achucarro that they want to separate the green for the yellow, which is very difficult, but it is possible to be done. And you have, this is different filter. It's very narrow for the green and very long for the red. So this is just to tell you that you have to choose the right filter in your protocol, depending on what you want to see. Okay. The recipe for the ACSF, and I think this is the last one. Let me see. Yes. So the recipe. This is something that you can find online. This is the one we use. Just to explain a couple of things. Normally, you will cut with this one, and you will register with this one. So you will cut your slices with what we call the ACSF sucrose. And why sucrose? Because here you replace the sodium with the sucrose. So in this way, you are minimizing the excitotoxicity from the activation of ionotropic channels. And also you use very high magnesium to block the NMDA receptors, which are excitotoxic glutamate receptors. And you are also using very low calcium for the same reason. So you are like protecting the slice in the cutting moment, okay? And in the regular is like the regular concentration of all the ions, no? Sodium is the main extracellular ion, potassium is the main intracellular ion, and you have phosphates to maintain the pH, and you have magnesium for the channels, for the NMDA, calcium, and bicarbonate. You have the bicarbonate is mostly to maintain the pH in the bubbling system, with the bubbling system, and glucose. Ascorbic acid is an antioxidant, and of course you have to adjust the pH. Normally I put the bubble here before, because if you add the calcium before adjusting the pH, the calcium will precipitate. Okay. And also the antioxidant, you can replace it with N-acetylcysteine or glutathione. This varies. This is very variable this part is variable from lab to lab. This is a recipe we use. If you write in Google ACSF acute slices, you will find very similar recipes. So that is all. This is the lab. These are our collaborators. As I told you, the lab is very big. There are many people. People working directly with me are these four. Maria actually is in another lab now, but she did most of the. She was the first author in the paper that we just got in eLife about the matrix and astrocytes. And if you want to check some of the macros, the script that we use, you can check in our GitHub page, or you can find us on Twitter and ask people. Usually they write and they ask for the macros, and I usually point them to the GitHub. But so I think that's it.